The central nervous system, CNS, which consists of the brain and spinal cord, plays a crucial role in regulating respiration, ensuring that the body receives an adequate supply of oxygen and expels carbon dioxide. The control of respiration involves several brain regions, including the medulla oblongata and pons, collectively known as the respiratory centers. Various respiratory centers involved are 1. Medullary. Respiratory center, dorsal respiratory group, DRG. Located in the dorsal part of the medulla, the DRG is primarily responsible for the initiation of inspiration, breathing in. Neurons in this group send signals to the diaphragm and external intercostal muscles to contract, resulting in inhalation. Ventral respiratory group, VRG. Found in the ventral part of the medulla, the VRG is involved in both inspiration and expiration, breathing out. While the DRG mainly controls the diaphragm, the VRG regulates accessory respiratory muscles and expiratory muscles. 2. Pontine respiratory group, PRG. Located in the pons, the PRG influences and modifies the activities of the medullary respiratory centers. It helps smooth the transition between inspiration and expiration and contributes to the overall regulation of respiratory rhythm. The apneustic and pneumotaxic centers are components of the respiratory centers located in the pons, which is part of the brainstem. These centers, along with the medullary respiratory centers, dorsal respiratory group and ventral respiratory group, play a crucial role in regulating the respiratory cycle. Apneustic center. Location. The apneustic center is located in the lower pons. Function. The primary role of the apneustic center is to promote inspiration, inhalation. It appears to have a stimulatory effect on the neurons of the dorsal respiratory group in the medulla, particularly those responsible for the inspiratory phase. Effect on respiration. Stimulation of the apneustic center leads to a prolonged inspiratory phase, resulting in deep and gasping breaths. However, it's important to note that the precise function of the apneustic center is not fully understood, and its exact role in normal breathing is still a subject of research. Pneumotaxic center, location. The pneumotaxic center is located in the upper pons. Function. The pneumotaxic center acts as a regulator of the duration and intensity of each breath, primarily by inhibiting the activity of the apneustic center. Effect on respiration. Stimulation of the pneumotaxic center results in shorter and shallower breaths. It plays a crucial role in preventing overinflation of the lungs by inhibiting the prolonged inspiration initiated by the apneustic center. The pneumotaxic center helps ensure a smooth transition between inspiration and expiration. 3. Chemoreceptors. Chemoreceptors, which are sensitive to changes in blood gas levels, particularly carbon dioxide and oxygen, play a critical role in respiratory regulation. Peripheral chemoreceptors, located in the carotid bodies and aortic bodies, detect changes in blood oxygen and carbon dioxide levels. Central chemoreceptors, located near the ventral surface of the medulla, are sensitive to changes in cerebrospinal fluid pH, influenced by carbon dioxide levels. Feedback mechanism. The central respiratory centers receive feedback from peripheral and central chemoreceptors. If there is an increase in carbon dioxide levels or a decrease in oxygen levels, these receptors send signals to the respiratory centers to adjust the rate and depth of breathing accordingly. Stretch receptors. Stretch receptors in the lungs and chest wall provide feedback on lung expansion. When the lungs are stretched during inhalation, these receptors send inhibitory signals to the respiratory centers, helping prevent overinflation. In conclusion, the regulation of respiration is a complex process involving the integration of signals from various receptors and brain regions. It ensures that the body maintains a balance between oxygen uptake and carbon dioxide elimination to support cellular function and maintain homeostasis.